So hi, everyone. Welcome to the second installment of our CSRC Summer Colloquium series specifically for students. Today's speaker is Ashley Schwartz. Ashley pursued her passion for mathematics and earned her bachelor's degree in applied math in 2019. Since then, she's continued her studies in applied math, and she just completed her third year in the computational science joint doctoral program between San Diego State and UCI. Ashley is advised by Dr. Udak George in the SDSU math department, as well as Carolyn Sant from the SDSU School of Public Health. And together they work in the very interesting realm of computational developmental toxicology. So her research aims to shed light on the potential risks posed by various environmental factors during early stages of life. Ashley's commitment to academic excellence extends well beyond her research. She's the incoming president of the SDSU SciAm student chapter. I'm very happy to be handing the baton over to her. And she's also honored as an ARC scholar by the SDSU College of Science. So welcome, Ashley. Very, very excited for your talk, and yeah, take it away.
Okie dokie. Thank you, Delaney, for the lovely introduction. Okay, I'm going to get my screen organized for a second and try and share this. So you should, oopsie. Okay, yeah, you should see the actual slides, right? Yeah. Okay, cool. All right, I'll get started. So hi, everyone. Yeah, thanks again for the introduction. As Delaney said, my name is Ashley, and I am a computational science PhD student. So today I'm going to be talking about a couple of projects actually related to my early graduate school work, where we use mathematical modeling to model embryonic development in the zebrafish model. And then, yeah, just a quick shout out again to my advisors, Dr. George in the math department, and Dr. Sant at the School of Public Health. We're really interdisciplinary in everything that we do. So all of our work together has just been really rewarding. So let's see, this goes, okay. To give a little bit of background and motivation between kind of all of the work that I do, as many of you guys may know, unfortunately, we come into contact with environmental pollutants every single day of our lives, whether that be in the water that we're drinking, just breathing it in through the air, or even the food that we eat, even the products we use, a variety of different things. These can pose a hazard to our health, not only now, today, but also during development. And that is really what I'm particularly interested in. So, of course, the human in utero environment is not readily accessible. We can't really study the way contaminants affect embryonic development because, of course, there are a variety of ethical concerns. So in developmental toxicology, we use zebrafish as a model for embryonic development. Zebrafish are really tiny fish that you can visualize that closely mimic human embryonic development. Surprisingly, greater than 70% of human genes have homologs in zebrafish. They are completely transparent. So as you can see on the screen here, if you look at them under a microscope, you can see all of their organs develop, which makes it really useful when we're studying the development of organs in general. And also they are fertilized externally, and they're making them minimally invasive models. So of course because they're fertilized externally, we can watch them develop, which is very different than say a mouse or a rodent model, where of course, you still can't access that in an in utero environment over time if you were looking at overtime investigations. So now that we know just a little bit of the big picture of what I'm interested in, talking a little bit about the current gaps in literature and where I fit into things. There are very few mathematical models for zebrafish development, and especially in developmental toxicology. They're just lacking currently at the moment. And we've recently stumbled upon a contaminant called TCPMOH, which I'll talk about in just a second, that is an emerging pollutant of concern. So really what I want to do is be at this gap, create mathematical models for zebrafish development that can help us understand what TCPMOH is doing during embryonic development, and then hopefully use these models to aid in the continual assessment of pollutant effects on development. So really at the intersection of all of these, understanding, what is happening in TCPMOH using the math model, and then using that model in the future. How can we leverage that in the future to be used continually as new pollutants continually emerge? So a little bit about what TCPMOH is. TCPMOH has been hypothesized that it can pose a hazard to embryonic and fetal development. It has been detected all over the world. So really off the coast of California, and then also it has been found in Japan and other areas, but it has been detected in coastal wildlife samples. So they've actually been detected in sea lions that have been, kind of ravaged with cancer, unfortunately. And there have been levels of TCPMOH detected in human breast milk. And then also it is structurally similar, and every time it is detected, it co-occurs with a legacy pesticide DDT, which, if you don't know what DDT is, it is a pesticide that was banned many, many, many years ago because it was very, very toxic to wildlife and humans. And it's kind of concerning that TCPMOH is co-occurring with DDT at such high rates. And we are starting to unfold kind of in our environment where these DDT, when it did get banned, where people disposed of these chemicals and how it's just been long lasting. So, of course, TCPMOH can pose a hazard to embryonic and fetal development. And because little is known about TCPMOH, we want to begin to characterize that. So my goal throughout these projects has been, in the early graduate school work that I've done, is to develop mathematical models for the assessment of environmental contaminant effects on embryonic development with TCPMOH as that model toxicant. And today I'm going to be talking about, of course, I'm going over now, I split it up into chapters. So today I'm talk to you a little bit about the introduction. I'll talk to you about the experimental methods and data behind all of the work that I will discuss. I will talk about a mathematical model that I developed to model the interaction between yolk utilization and fish growth in the zebrafish. And then I will talk about a mathematical model in the form of a network model approach that identifies temporal patterns of structural malformations that were seen following exposure to TCPMOH. And then lastly, I'll just bring it all together, talk big picture about what these models bring to the table. So again, let's get into the experimental methods and data. So I wanted to talk about this coming from a math background. I had no experience in the lab, and when I came into my graduate school career, I really wanted to get an interdisciplinary viewpoint on things. So I got into the lab and we ran these experiments for these studies. And really how it starts is we collect embryos from the breeding tanks. And here are just some pictures of this is actually our old zebrafish lab. It's newer and bigger now, but this is a photo of our zebrafish tanks and what the zebrafish look like in the tank. We start off by collecting embryos from the breeding tanks, and then we put them into exposure groups or experimental group solutions. And each zebrafish is pretty tiny at the embryonic stage. They each live in a 96 well plate. So in this case, we have 5 different exposure groups. We'll have the control group, which is a DMSO solution, and then we have 0.1, 0.51, or 5 micromolar TCPMOH. And each exposure group will have about 20 to 38 embryos per exposure. And then these were tests that were run during the developmental stage only, so just on those embryonic zebrafish or the zebrafish embryos. Throughout the developmental stage, we capture microscopy images to actually track that development process. So we'll take an image every single day post-fertilization. And to do this, we actually remove the zebrafish from the well plates and stage them to make sure that they are perfectly straight, as you can kind of see here. And then we'll take those images. And from those images, we will assess morphological data, mortality, a variety of different endpoints that I'll be talking about today. So again, zebrafish are really great because we can visualize all of those organs develop through that microscope, and we can assess proper development of them over time, which is really important because we're doing a continuous time investigation here. So the first thing I want to touch on is a little bit of the supporting data and the results that we found from these experiments without looking at the math modeling yet. Survival, TCPMOH significantly increased mortality, decreased survival. So we saw that the high exposure group, the 5 micromolar group experienced, and the 1 micromolar group experienced nearly 100% mortality by the end of the developmental stage. And as you can see here on the graph, all these little stars just mean statistically significant. So even in the lower, the 0.5 micromolar group, we're still seeing mortality towards the end of that developmental stage, but the 0.1 micromolar did not experience mortality. We also assessed fish length, and embryos exposed to the 5 micromolar TCPMOH group experienced that stunted growth statistically significantly, and the others did not experience such a big difference. But you can see there is still a difference in that growth over time, which was particularly interesting. From that, in developmental toxicology, we often assess a variety of embryonic morphological endpoints. And I'll be talking about these a lot today. So just to go over these, I'll talk about pericardial edema. This is essentially an inflation of the sac surrounding the heart. So this is a normal fish here, and this would be an TCPMOH exposed fish, an abnormal fish. And in this case, you can see there's a lot of fluid in the heart, and that really puts pressure on the heart and makes it hard for the zebrafish to continue to develop. Yolk edema is essentially inflation of the yolk sac, which is the primary nutrient source, which can alter nutrient uptake. The swim bladder inflation, this is a normal fish with the swim bladder inflated. This is when the swim bladder doesn't inflate. This can cause issues because if the fish can't swim, once the development ends and the fish needs to be able to swim to eat, they can't do that. And then kind of same thing with the craniofacial malformations, that it makes it harder for the fish to consume food once that developmental stage ends. And then just to bring that up again, during the developmental time point, of course, the fish can't eat. This digestive system hasn't been developed. They get all their nutrients from this yolk sac here. And I'll bring that up again in just a second. So now that you kind of get an idea about what kind of data we're looking at and the different things that we've assessed or endpoints that we assess during those developmental toxicology endpoints, the first thing we thought was, how can we model this temporally using a mathematical model? And is there any relationship between yolk utilization and the overall fish growth in the zebrafish? And that's kind of where the first step of my project took place. So really the objective of this chapter, now going into chapter 3 here, is to develop a mathematical model to represent the interaction between nutrient absorption and fish growth in the zebrafish model. So figuring out how we can model this using differential equations. I wanted to identify key developmental windows to kind of aid in developmental toxicology studies. Oftentimes, we only look at 1 or 2 time points. So even reporting on those 7 time points that I just showed previously is very rare in developmental toxicology studies. Typically, it'll just be around at the end of the developmental time point, or maybe just before, a certain stage. So we wanted to assess what kind of developmental window was critical for nutrient availability, because if toxicology studies are only looking at 1 time point, even if we continue to do that in the future, what time point is the most important? So those are kind of the questions that I wanted to get at. So again, the 2 metrics that we are looking at here are the overall fish length, which is just taken as a measurement from head to the end of the tail. And then the yolk sac is this protruding yolk sac here. Again, this is the nutrient source for the fish, and we would capture the area of this to determine the amount of nutrients available for the fish. The nutrients that are available, the yolk sac is made up of a variety of nutrients, whether that be lipids and micronutrients, and they just essentially sustain metabolic function during the developmental time point. This is kind of similar to how humans get their nutrients through the placenta. All the nutrients come from here in the zebrafish. They have that finite amount in the beginning, and that has to last them essentially until they're able to eat at the end of the developmental stage, which is important as I continue to talk about the modeling practices. So the other thing that I want to talk about is with the data that we used for this particular study. I will be talking a lot about the control group in this particular portion. We wanted to characterize a normal behavior of nutrient absorption and fish length, and then bringing it back to TCPMOH, seeing how that's altered once the model has been established in the control environment. And then also, because yolk edema, so in one of the images that I showed earlier, because yolk edema inflates the yolk sac, it makes it really hard to actually capture a measurement of the area of the yolk. So we just used the low exposure group, which had the least amount of, well, also survival. So we wanted to make sure that we could continue the, oh, sorry, it flew. I just stepped on my dog, I apologize. We wanted to be able to make sure that we could track the same fish over time. So survival and edema, we just use the control, and then the low exposure TCPMOH group. Okay. So again, we're looking at a control environment here. So we wanted to build a mathematical model and then test the model fit. So to do that, we use parameter estimation, nonlinear least squares regression analysis. In this case, of course, we want to minimize the error between the data that we found and the model itself. And this is just done very typically using this optimization function. And then also the optimization algorithm that we used was just a constrained optimization algorithm. I believe it was a convex interior point algorithm. And then from there, to assess model performance, because we tested a variety of different models, we just used a very basic R-squared metric, which is of course just a measurement of how well the model fits the data using the sum of squared residuals and the sum of squared errors and the sum of squared total, which is just the formula is here on the screen if you were curious. So the first step was really to model both yolk utilization and fish length independently. Sorry, it's being very crazy. So to do that, we wanted to look at a variety of different models and see which kind of behavior fits the data best. We wanted to consider a variety of different biological restrictions that kind of play a role into choosing the proper model or even investigating the right model. Based on the data that we've seen and then or the behavior of the data, we looked at an exponential decay model, a decreasing Hills function, and a logistic decay model. And these are just the model equations here on the screen. From that, we performed, of course, nonlinear least squares analysis on all of those to assess the model fits. And as you can see from the R-squared, the best model in this case was the logistic decay model. And then on this right-hand side, you can just see this is the normalized yolk area over time for each of the samples. These are all the control group samples or the control group samples over time. So the model, so we can conclude here that logistic decay represents the nutritional absorption over time in the zebrafish. And then we can perform a very similar analysis for the fish length. So again, using based on the shape of the data, we thought that maybe the Hills function or the logistic growth model might work for this case. And they were very similar, which is something to note. Obviously 0.98 and 0.9899 are not very different. But as we begin to scale outward into the interaction model, the consistency between the yolk and the fish length was advantageous. And of course, logistic growth is a pretty typical model for growth, biological growth dynamics. So we did want to stick with that for those reasons. And you can see here again, the model does really fit the data well. It's a very simple logistic growth model here. I want to also say that even though this is rather simplistic in some ways, this has not been determined in zebrafish yet. So we didn't really know the behavior or the dynamics of the fish length or the yolk utilization over time. So even this independent investigation was very impactful for the community to understand how that behavior occurs and even looking at different windows for when those growth spurts occur. So as you can see here, if you were really looking at, there's not much growth changes after day 5. So if you're looking at a day, if you're trying to investigate fish length, maybe looking at day 5 would be sufficient because you don't need to continue on your experiments for more days because you're not really going to see much of a difference. So going forward, we wanted to build an interaction model or really build the OD model kind of predator-prey style. So what we did is we assumed biologically from our biological hypothesis is that nutrient uptake will fuel both fish length or the overall fish growth, and then also organogenesis within the body, so the development of other organs. And we use that to kind of build the OD compartmental model from there. So for yolk utilization over time, we have yolk utilization elsewhere in the body, and then also that yolk utilization that feeds fish length. And in this case, this is taken to be logistic. The initial condition here is just the initial yolk size at the beginning of the developmental stage. And then similarly for fish length, we just have a logistic growth of fish length that is proportional to the yolk absorption at time t. And here are just some model parameters in case you're interested of those, but really we're focused on, these rate parameters. What is that rate of yolk utilization? What is that rate of fish growth? And how are they proportional to one another? This model does fit the data quite well. Again, this is the mean data plus the standard error from the mean. And then the black line is the model fit. And the model fit is taken to be with the mean parameter values from the nonlinear least squares analysis. The R squared here was 0.98. So again, it does fit really well, which was exciting in some ways. We were able to represent this interaction pretty well. And from this, we can kind of do some more investigation of the model to see what else we can find, not only, confirming biological relevance, but getting any additional information. So just a quick stability analysis on these things, just to kind of fact check. Obviously, stability analysis on simple logistic equations are pretty simple, but confirming the biological assumptions here. So stability analysis is, of course, to determine what the function or what the behavior is going to occur, what kind of behavior is going to occur in the long run. And in this case, we have two steady states, steady state 1 and steady state 2. This is just essentially 0. It just means the yolk is 0. And then also K1 is that initial yolk size. So we found that SS1 was stable and SS2 is unstable. This implies mathematically the yolk area will ultimately be depleted over time, which does fall in line with that biological assumption. So if you remember, I said previously, there is that finite amount of yolk during the developmental stage. At the end of development, when the fish is able to eat, they no longer rely on their yolk to grow or even just live. So that does fall in line, which is good. Similarly to fish length, this is very similar. In this case, we have fish length equals 0, and then fish length equals its maximum. And in this case, the maximum fish length will be reached at the end of that developmental time point, which again falls in line with that biological assumption. It is important to note too, just a reminder that this mathematical model only does apply during that initial time point. So during that developmental stage, embryonic development specifically, and does not extend into the juvenile stage, because in this model, we are assuming that all nutrient availability comes from the yolk, and the yolk does not exist in the juvenile stage of the fish. For the interaction model, again, very similar stability analysis here. The first steady state is 0, 0, which is a non-physical point, especially because the fish length is never 0. And the second second equilibrium point, can't keep my words right, is over here, is represented by this open circle here. And following stability analysis, Jacobian linearization, all of that kind of good stuff, we found that that is an unstable saddle node. And if you follow the solution trajectory here, it kind of makes sense. We start with a full yolk at essentially its maximum, and then eventually we end up meeting the length maximum towards the end of that developmental time point. So mathematically speaking, the model does fit the biological assumptions, which does confirm its applicability to this biological application, which was good, so that we can move forward with confidence in any sort of implications the model results in. So looking at another thing we are interested in is, as I said previously, what is that developmental window that's important? How does nutrient absorption have an effect on overall growth? And we first did a very simple statistical analysis looking at, is there any correlation between the change in yolk area and the change in fish length over time? So we just took for each sample, what is the change in yolk area? What is the change in fish length? Plotted it here, did some statistical analysis, and we found the only significant day was actually days 4 through 5, between 4 and 5. So we found that between day 4 and 5, there was a statistically significant correlation between the change in yolk area and fish length. And we took that a little bit further using the model. And here I just plotted the rate of yolk utilization over time. So the differential equation model. And by finding its maximum, we found that the maximum rate of yolk utilization occurs at 3.5 days post-fertilization. So what's interesting is that we're hitting our maximum of yolk utilization at approximately day 3.5, but we are also seeing this correlation between days 4 and 5. So we began to think, what might be happening? What is that really critical window? Is there a delay between the time that nutrients are absorbed and the fish is ultimately able to grow? So we did take that a little bit further and wanted to look at that. So looking at, we use delayed differential equation modeling. So what is delayed differential equation modeling? Delayed differential equation modeling essentially uses two different time points. So we have time t, and then we have time t minus tau, where tau is some time delay. So in this case, we assumed that the yolk utilization just occurs at time t, and then by the time it takes to fuel fish growth, there's a delay in that. So that was the assumption here, and this was the model that we developed for the delayed differential equation model. And again, using nonlinear least squares analysis, we wanted to determine if you fit this model and tau is 0, that means that there really is no delay. But we did find that tau equals 0.5 did fit the model best, which means that there is a 12-hour delay between yolk consumption and ultimate fish growth. So this was particularly interesting, again, pulling back to what I just talked about previously. Yolk utilization reaches its maximum at 3.5 DPF, days post-fertilization, but there could be a 12-hour time delay to when that yolk utilization actually fuels fish length and overall growth of the fish. So it doesn't occur immediate, which again, biologically speaking, does make sense. So now that I have all that out of the way, I can talk a little bit about TCPMOH and what we saw in the exposed case. So again, we're looking at the 0.1 micromolar TCPMOH group. All I did here was fit the model to the TCPMOH data. So the goal here was kind of twofold. One, making sure the model is robust enough to withstand an environmental condition and increase variability in the samples. And then two, use the model to predict if there was a change in rate constants for the exposed group. So in this case, I have the normalized yolk area, black is the control group, and blue is the TCPMOH exposed group. And you can see here that there is an increase in variability as seen by the error bars in the TCPMOH group, which really just, of course, means that some were greater, some were higher, but there was definitely, especially in the TCPMOH group, there were some that had really low fish length and some that were kind of average. So even with that variability, the model still fits the data quite well. In this case, the TCPMOH group, it was still a 0.91 fit for, again, this is showing the mean fit, but we did the fit on each individual sample. And from this analysis, we found that TCPMOH increases yolk utilization very slightly, as you can see by this dip here. This rate does occur faster, essentially, in comparison to controls. And then we also found that TCPMOH decreases the fish growth rate. So you might be wondering, oh, well, they're using nutrients faster. Isn't that better? In the zebrafish case, not so much, because again, they only have a finite yolk source. So as the fish continues to use their yolk faster, they're essentially malnourishing themselves in some ways. And that could potentially be what's leading to this decrease in fish growth for the TCPMOH case. Because again, once the yolk is gone, they have no way to get nutrients, and maybe they're over absorbing them and not able to actually metabolize and continue to use that to grow. So definitely some more investigation is needed there on the biological standpoint, but the model is very promising in understanding these differences between the control and the exposed group. So just some key findings on this chapter. First of all, again, this is the first mathematical model to describe nutrient absorption and fish growth in the zebrafish model, both independently and as an interaction. So definitely some feedback that I've got from this model is it's very useful in the simplicity as well in the interdisciplinary setting. So really being able to be applicable to a variety of different studies in the developmental toxicology space, as it can be definitely used to understand differences in new emerging contaminants. So I'm actually presenting on TCPMOH here, but we have used this model to study a variety of, well, one other chemical that we did publish on, which was interesting as well, and that is a PFAS chemical. So it's definitely extendable to different environmental pollutants. The developmental biology application, again, we found a key developmental window from where the yolk utilization reaches its maximum, which is very important as we think to actually, design our toxicology studies and developmental biology studies, and understanding like when yolk utilization occurs at its highest rate, so that we can kind of just better design the studies. So of course, if you expose your zebrafish well after this point, maybe you're not going to see any difference in that nutrient uptake because you've exposed it well after all the nutrients have been absorbed. And then again, when it comes to TCPMOH, we did find that TCPMOH has an increase in yolk utilization and that decrease in overall fish growth, which is picked up by the mathematical model. So moving on to the next chapter. So again, all of this work is under the same umbrella of the same experiment. And what we saw in the beginning was there were a variety of different malformations that were occurring, or abnormalities in the zebrafish. And we wanted to see, we did see some patterns over time, and we wanted to see if there was any relationship between the abnormalities that were occurring. So instead of looking at them independently, how do they co-occur, and how does that potentially lead to the presence of further abnormalities in the future? So this project, titled Dynamic Network Model, identifies temporal patterns of structural malformations in zebrafish embryos, takes a mathematical modeling approach from a network modeling perspective. And I'll talk about kind of all of that stuff from a very novice standpoint. So again, the objective here is to identify the temporal patterns. So looking at that temporal aspect of abnormality is not just looking at them at one time point. How does the occurrence of one in the future, in an early time point, lead to something in the future? And how that might relate to mortality. So those are kind of the questions we were getting at. And then again, I want to refresh you on some of these morphological endpoints because I will be talking about them again in detail. Pericardial edema, yolk edema, cranial malformations, jaw deformity. We have spinal deformities, and then also swim bladder inflation. And then mortality, which of course just isn't pictured here on the screen. For this project, we used the 0.5, 1, and 5 micromolar TCPMOH group. We were interested in malformations, and just because of that, we left out that very low exposure group that didn't really have much of that here. So the first step in all of this was to determine, was there any relationship between the abnormalities at all? So kind of did a big statistical test here. This is just a, statistical analysis of each individual abnormality pairs. So between pericardial and yolk sac edema, is there an association between those two abnormalities at that time point for that exposure group? So this is just a heat map showing the p-values of association for all of those pairs using Fisher's exact test. So these are just binary abnormality outcomes, which of course lends itself to Fisher's exact test versus a different correlation coefficient such as Pearson's or a variety of the other ones that are out there. And from this, it's very overwhelming. Like, what can you actually get from this? But when you have a lot of data like this, it really lends itself well to network modeling. And I'm going to talk about how you can how we developed a network model based on this data. So I'm talking about a dynamic network model here. And what that means is a network model that has values that change over time. A network model is really just a mathematical graph. And in this case, it has a set of nodes, links, and a mapping function. The nodes in our case are going to be the actual abnormalities themselves. So in this one is yolk sac edema, pericardial edema, and so on. The links themselves are going to actually be weighted, which means they have an associated score, if you will. And in this case, that comes from the p-values of association that I just showed on the previous screen. So if we compute the weight as 100 times 1 minus the p-value, that ensures that a really significant association is given a large weight within the network model. So again, if yolk sac edema and cranial malformations are highly associated, they will have a strong link. So this weight here would be associated to that link. And then every network has what's called a mapping function. And in this case, the mapping function is an adjacency matrix that essentially is just a matrix representation of all of these links and weights. So it'll be a square matrix where each column and row represents a different node, in this case, a different abnormality, and each matrix entry represents that link, in this case, the weighted link. So that's kind of how you represent it computationally as well. And it lends itself really well to a computational investigation because, as you all know, if you've dealt with a lot of things, matrices are good and bad sometimes, but in this case, they're good. It's not too complex. We're not very sparse. So it lends itself well to this particular investigation. And then again, just to reiterate, these change over time. And so that means that, as you saw on the previous screen, we do have our p-values are changing over time, and that is what, sorry, my dog just barked, it's kind of distracted me. Anyway, and then also we have a network model for each exposure group. So we have a control dynamic network model, the 0.5, the 1, and the 5 micromolar group. And we have an individual network over time for each of those. So that's kind of the model structure and how the model is developed. And then from there, we can actually analyze the model. And to analyze the model, we use what's called centrality analysis. Centrality analysis is essentially a way to get network topology from a network model. In this case, centrality is a score that's given to the network that assigns a ranking of co-occurrence between the observed abnormalities for the different exposure groups over time or the different networks that we're dealing with. So it's a really great way to kind of just get some information from the networks when you have a lot of data that you're dealing with, as you saw in that heat map. And there are two different centrality scores that we looked at for this particular model. The first one is what's called degree centrality. Degree centrality is really just a measure of, well, if it wasn't weighted, it would be a measure of how many connections each node has. In this case, it's the sum of all the weights of all the connections for each node. And then when we talk about a global centrality or a maximum degree centrality, that's just taken to be the maximum degree of the network. So each node will have an associated degree, and then the global degree centrality score is that maximum degree for the network that kind of represents the overall connectivity. Another centrality score we looked at was eigenvector centrality. Eigenvector centrality is essentially found by taking the eigenvalues of the adjacency matrix, and the global eigenvector centrality is taken to be the spectral radius or the maximum eigenvalue. Eigenvector centrality is very similar to degree centrality, except it also includes transitive influence. So what that means is it's not only considering, so degree centrality just cares about the direct links to a node. Eigenvector centrality will incorporate the next level, so it's neighbor's neighbor. So if you have a node that is very connected immediately, and then also its connections have a lot of connections, that node will be given a stronger score. So globally speaking, when we did this for each network, for each exposure group, we did degree centrality here. So this is the normalized maximum degree. So this is just the maximum degree over time for each exposure group, the control 0.5, 1, and 5. And then what's interesting here is that, well, that you'll notice, first of all, the control group has no centrality scores. This falls in line with that heat map that I showed previously, because if you notice, there was no significance between the co-occurrence of abnormalities in the control group. And the 5 micromolar group had a really high centrality score at 2 dpf, but then kind of stayed stagnant and lower than the 1 micromolar group. And this was actually the same thing occurred in the eigenvector centrality or the spectral radius score. So why, you might ask? Well, in the high exposure group, if you remember from the very beginning, mortality occurred really quickly. Mortality started to occur right at 2 days post-fertilization, which is basically the fish had a day to develop and it had already died. And when we actually assess morphological endpoints, if the fish has died, we wouldn't assess any other abnormalities because it's just impossible to really know, because I know many of you haven't seen a dead zebrafish, but trust me when I say you just can't know. So we don't assess any of those abnormalities. So you can't really determine if there was any co-occurrence between anything. So there is a bit of survival bias when you're taking this modeling approach, and just something to be aware of in how these scores are coming out and kind of why it makes sense, why those centrality scores are lower. Because mortality occurs so early, the fish has no ability to continue to develop abnormalities and therefore has no co-occurrence of abnormalities beyond mortality. The 1 micromolar exposure group did experience mortality, but not till the later time points. So a lot of the fish that did die did experience abnormalities previously, and we can kind of see that in these centrality scores here. So we can determine there's definitely a difference between these exposure groups. We can see that the 1 micromolar exposure group is definitely significantly has a higher co-occurrence than the 0.5 micromolar group, and then especially different than the control. But we were also interested in what abnormalities were causing these scores and what abnormalities were the ones that are kind of driving the connectivity of the networks. And in this table here are just the abnormalities that correspond to the maximum degree or the maximum spectral radius. And in most cases, you can kind of just see populated throughout this table. Pericardial edema and yolk sac edema were consistently identified as the predominant nodes, or the nodes with the most influence, or the nodes that were corresponded to the maximum centrality score. There were definitely some other abnormalities thrown in there, like cell deformity over here in this low dose group. But for the most part, pericardial and yolk sac edema were definitely most apparent. And from here, we also wanted to look at individual node centrality. So looking at all the nodes over time. And that's what this is here. So this image over here on the screen is the low dose group, the medium dose, and the high dose, exposure, low exposure, medium exposure, and high exposure group. And in this case, we plotted the centrality score over time for each of the different nodes in the model. And what this showed is that even though, spinal deformity had the highest exposure or the highest centrality score here at 7 days post-fertilization, pericardial and yolk sac edema were not far behind. And then also, if you look at this graph here, this is essentially just showing all the connections over time. In the 0.5 micromolar group, yolk sac edema and pericardial edema were the only abnormalities that co-occurred at early time points. And overall, in the entire graph here, anytime there is co-occurrence between abnormalities, yolk sac edema and pericardial edema were always co-occurring. So all of these factors really pointed us to believe that yolk sac edema and pericardial edema definitely are having a large impact on the presence of abnormalities that occur later on, because they are always occurring at the early time points and then continue to co-occur as more of abnormalities form in the later time points. And thinking about what I want to say next. So another thing too that's important to say here, pericardial edema and yolk sac edema are special abnormalities, I will say, in zebrafish. When they do co-occur, they're hallmarks of what's called blue sac disease. Blue sac disease is a disease in zebrafish that essentially you get yolk sac edema, pericardial edema. It's just the hallmarks of it, or you get yolk sac and pericardial edema, and then the fish dies at later time points. This is a very typical disease that occurs in chemicals from the PFAS group, which, if you don't know about PFAS chemicals, they are chemicals commonly found in non-stick products or a variety of other products that you come into contact with, and a lot of them have been known to cause cancer. So as in zebrafish, if the zebrafish is exposed to a lot of these PFAS chemicals, they get blue sac disease. These zebrafish were exposed to TCPMOH. They're showing signs of blue sac disease. So it's definitely concerning on that TCPMOH side. And this model is really good at picking up on that co-occurrence to kind of inform something that we should be watching out for, or something to, point out to the community that needs more investigation in. So some key findings and contributions for this project. We have this dynamic network model. This is kind of the first, in this case, it was a development of the first dynamic network model for developmental toxicology, looking at abnormality co-occurrence over time. Definitely the first one to kind of take a look at this. And I believe it'll be useful in the future, looking at new chemicals and really understanding how those relationships continue to occur and how they differ with a variety of different environmental contaminants. When it comes to developmental toxicology and TCPMOH in general, yolk sac and pericardial edema seem to be the predominant abnormalities co-occurring. And then also when it comes to TCPMOH, not only is it the yolk and pericardial edema are occurring, but also TCPMOH exposed samples are experiencing an increase in abnormality co-occurrence across the board. So yeah, I'll continue to just wrap up a bit here. So just to kind of reiterate again, some key research findings of everything that I talked about today. The first part, when I talk about the materials and methods, in that study, which I didn't talk about in depth with that study in general, but we found that TCPMOH increases the incidence of embryonic abnormalities and mortality in the zebrafish model using just like a basic statistical analysis. And then in chapter 3, I showed you guys a developed mathematical model that identified the critical developmental window for embryonic nutrition and growth in the zebrafish. And we also found that when we extend the model to TCPMOH, we identified an increase in yolk absorption and a decrease in overall fish growth. And then in chapter 4, I talked about how dynamic network modeling, using dynamic network modeling, I identified yolk and pericardial edema to be critical abnormalities, potentially causing further abnormality co-occurrence. So they always occurred early on. They could be leading to those further abnormalities, including mortality. And then TCPMOH caused that really strong co-occurrence between pericardial and yolk sac edema, and also has a higher co-occurrence of abnormalities overall when compared to controls and between exposure groups. So with all this being said, we can definitely conclude that TCPMOH impacts the nutrient availability for the zebrafish, and it is seeming to have an issue with the zebrafish being able to metabolize the nutrients and then develop properly. So it's definitely of concern and definitely needs more investigation on that end. And just to talk a little bit also about kind of where I'm going with this research. So again, this is kind of some of my early graduate school research and really gave me experience in the interdisciplinary setting. So again, actually running the experiments, actually developing mathematical modeling approaches for experimental design, realistic experiments. And I think that's a really important tool that I gained as an interdisciplinary researcher through these projects, is understanding what is realistic for a mathematical modeler, what kind of data can we get, and how I can use that to inform things in the future. And then also computationally speaking, learning how to develop these network models and how I can extend those outward. So where am I going next? If you happened to hear or see me talk at the, or watch my video at the Access event, I talked a little bit about how I've already, or some of my recent work, I've been extending to more complex data sets for zebrafish, mainly focusing on RNA sequencing data sets and looking at the transcriptomic response. So the gene expression responses following exposure to a variety of different chemicals. And looking at more of a multi-layer network approach. So not only incorporating machine learning and other techniques on the bioinformatics, but then extending outward into a multi-layer network modeling approach for a variety of different things, which TBD in progress. And then also extending a lot of this mechanistic data into the community. So using the same modeling approach of multi-layer network modeling to utilize community adverse birth outcome data to investigate and link it to the mechanisms of toxicity we're seeing here. So extending it out into the community, into California, where there are tons of data on pollution burden, birth defects seen in the community, and how these all link together, all using network modeling, in this case, a multi-layer network modeling approach. So yeah, that's kind of where I'm going with my dissertation. Point out some references here. These are the 3 publications. Well, these are 2 publications. This is a recently submitted manuscript, or that's currently under review, that I essentially describes chapter 2, 3, and 4, all the chapters that I talked about today and how they all relate to TCPMOH. Actually, sorry, again, this one actually reports on a different chemical, but anyway, I talked about the TCPMOH application here. And yeah, that's basically it. So thank you. I'm happy to take any questions. Again, I just want to say thank you to my advisors, Dr. George and Dr. Sant, and then a few of the Sant Lab members that actually helped me run the experiments and do the chemical exposures and all that kind of good stuff. And the following institutions for the funding and support over the years. So yeah. Thank you.
Good job, Ashley. Does anyone from the audience have a question? I actually have one. Maybe I can start. So your research is so interesting and very foreign to me on the physics side. But you were talking about like, furthering application of that developmental model and potentially adding like multiple layers. I was just curious, like taking that like type of model that you have and expanding it to different types of like chemicals or toxins. What does that actually look like? Like, do you need different types of data? Do you need to restructure the model? Or is it just kind of like a good baseline, you can expand on it?
Yeah, so from the dynamic network modeling approach that I talked about, that specific model can definitely be expanded to investigate other chemicals individually. When I'm talking about moving forward, I'm talking about kind of using a lot of the tools that I've learned in the network modeling to extend to a multi-layer network approach, and kind of switching gears on the data, mainly because the data that I need to use in the future there has to be more of it as we start to extend. So that is definitely a challenge, I think, as a math person coming into this space and coming into the experimental space. Is data is always a challenge, and especially as a computational scientist, you want as much data as you can get. So I'm kind of moving into bioinformatics or RNA sequencing data. So you'll get a lot more data that way. And that's kind of what I presented on in the Access event. And I do have like one slide here, which I can kind of just because this is just a picture. But in this case, you can see that in this case, we're looking at a variety of different chemicals. So this one has like 35 different chemicals. And then we're looking at this one is just showing the genes involved with pancreas development, but we can look at all the genes, and that's over 30,000 different genes. So instead of looking at just morphological endpoints, extending it into kind of genetic endpoints or gene expression endpoints rather, and looking at the mechanisms of toxicity. So what I mean by that is kind of on the molecular side, the molecular biology side, what genes are expressed, and then how does that relate to the phenotypic screening that we have. So it's a mix. It's a data shift, but hopefully they'll all kind of come together, because the idea is that, hey, I know that this chemical is really leading to these deformities. And I also know by this investigation, this chemical is leading to these gene expression changes. And this chemical does this, and this chemical also does this. How can we bring that all together to kind of make better informed decisions or predictions about emerging pollutants? So thinking about, okay, this is the data that we have. This is the data that we don't have. This is the chemicals that we've investigated already. These are the chemicals that we want to know but don't run the experiments on. How can we use our models to build those predictions? So kind of a long-winded answer, but it's an extension, different data, and all that kind of good stuff. So
no, I appreciate it. Yeah, and thanks for showing. Any other questions?
Yeah, I've kind of a dumb one. Actually, do you do the wet lab stuff for this too?
Yeah, so I did that for, yeah, I know, scary, right? I know, I was like, what the heck? That's what, I did do the wet lab experiments for the TCPMOH study. So I was definitely in the lab, learning how to pipette up a zebrafish and all that kind of good stuff. So it was a complete new experience. But it was, like I said, very useful, because it really gave me the idea of how difficult it is, especially for these continuous time investigations. I mean, we would spend at least an hour imaging every single day. And then, we would do the chemical exposures, we would mix the solutions and then re-expose the fish. And it takes a long time. So it's like, when people ask me, why don't you have more samples? I'm like, if only we could, right? So it's just a matter of that balance. But yeah, I did do the wet lab experiments on this project. I haven't been in the lab and ran any other experiments in a while, but yeah, for this one I did.
Does George get lab space for that? Or did you?
No, so that's all through Dr. Sant. So she's the one who has the zebrafish lab. in the School of Public Health, and that's who she's like, that's her lab. And they run a variety of different experiments for, I mean, obviously this one was on TCPMOH, but they're consistently running experiments on a variety of different things. We're always working, doing things, but that's what she does. So she has a lab space in the Bioscience Center with a big zebrafish room, regular wet lab space, all that kind of good stuff.
Yeah, I got banned from doing things with pipettes in my bio lab. And I ended up with math in the first place because I was so bad in the lab.
That's so funny. Yeah, it's definitely a skill that's scary, especially when you're pipetting zebrafish. You have to pipette them, and if you don't do it properly, you can break their backs and ruin the study. So, but anyway.
Okay, cool. So last call for questions, anyone? Okay, if there's no other questions, I'm gonna thank our speaker again. Thank you, Ashley. Again, great presentation.